Dear students, I welcome you all to the 13th session of Reproductive Biology and Developmental Biology paper prescribed by the Mangalore University for 6th semester BSc Zoology. In this session, we shall consider the branches of embryology and the scope of embryology. Let us first look into the different types, different branches of embryology. The embryology, which is a study of the development of a fetus or embryo, includes several branches. This classification is based on the kind of study that is being undertaken during the detailed analysis of the development of a fetus. The first such branch is the descriptive embryology. Descriptive embryology is the field of embryology which is associated with the morphological description of different embryonic stages in the ontogenic development of individuals of different species. This includes the initial works of embryologists till the 18th century. So if you have seen the 11th session of the same chapter, we have studied a detailed account of the history of embryology, where we came across with many scientists such as de Graaf, Wilhelm Roux, Anton von Leeuwenhoek, August Weismann, William Harvey, etc., who gave extensive contributions to the descriptive embryology, where they tried to explain the different stages of the development of embryo with the help of the morphological descriptions that are derived out of the study of the external features of the embryos, like how it is depicted in this diagram. You can see how the development of the human embryo takes place from first month till the 9th month, or depiction of fertilization as well as cleavage, blastocyst formation and implantation of the human blastocyst to the uterine wall. So all these kinds of works comprises the descriptive embryology. In broad terms, descriptive embryology is the study of the different embryonic stages using morphological descriptions. The next branch that we are going to study is the comparative embryology. This comparative embryology is also a branch of embryology that deals with the comparative study of the ontogeny, the embryogenesis of animals belonging to different groups. As it is shown in this photograph, that we can see different stages of development of 5 classes of vertebrates: the fishes, the amphibians, the reptiles, the birds, and the mammals. Here you can see that the initial stages of the development of all vertebrates is almost similar. That means During the embryogenesis, almost all the embryos look alike. This is probably due to the common ancestry that we all share with the other vertebrates. So this kind of embryology, the comparative embryology, helps us to compare the different stages of development of different groups of animals. This can also be done to the invertebrates as well. You can look into the developmental stages of different invertebrates as well and compare the differences in their ontogeny as well as in the developmental pattern. But by and large, the developmental process of animals from coelentrates to chordates, the mammals, is almost similar. It starts with a single-celled fertilized ovum called zygote, and through series of cleavages with certain differences among different groups, they reach a stage called blastula. Then the formation of germinal layers takes place due to the movements of cells, which we call the morphogenic movements, about which we are going to study in detail in the coming sessions. And the gastrula is formed, and then gastrula moves out through morphogenic movements to produce different organs and finally the entire animal. So this process of development as well as pattern of development is almost similar among all the animal groups. So the comparative embryology allows us to compare the developmental process across the different groups of animal kingdom, from which we can decipher the probable evolutionary history of the organisms. Later on, the embryology shifted towards the experimental processes, because until the beginning of 18th century there were no experiments concerned with embryology. Many scientists have worked on the field, but they were concentrating more on the patterns of development as well as the morphological features of embryos rather than experimenting with it. But the scientists like Spearman and Mangold and many others, including Wilhelm Rauss and Child as well, started many experiments with certain organisms such as Echinus, a kind of echinodermate, and Xenopus and salamanders, which are amphibians. By definition, experimental embryology is the study that attempts to understand various fundamental mechanisms in the development of different animals, like fertilization, cleavage, gastrulation, embryonic induction, determination, differentiation, etc., using experiments. So these are the two important depictions of experiments that were carried out during the early days of embryology. You can see here the fertilized egg of the salamander was bifurcated using a thread, but both the blastomeres had a part of gray crescent, which ultimately gave rise to two normal embryos. But if you allow the blastomeres to not to have the gray crescent, then ultimately that part of the zygote which did not share the gray crescent region, did not develop into an embryo. Instead, became a simple mass of tissue. But in another experiment, this was carried out by Wilhelm Rauch, where he separated the 4 blastomeres at the 4-cell stage after second cleavage, and he looked at the developmental patterns, and all the 4 cells gave rise to the entire animal. But if you allow these 4 cells to remain contact, then they produce only one larva. So this experiment was carried out using the Echinus zygote. So here we can clearly see the differences between the process of development, where if gray crescent is absent in a particular blastomere, and that particular blastomere failed to develop into completely functional animal. So this kind of experiments were carried out during the initial days of embryology, and they came up with many wonderful results, as well as the experiments carried out by Feynman and Mangold ultimately took them to the base of Nobel Prize too, about which we have studied in detail in the previous session. For more information about the experiments of Feynman and Mangold, you can refer the 12th session in the same paper, or you may also refer the 22nd session and 23rd session of the same paper. The latest field in the field of embryology is the chemical embryology. So this is a branch of embryology that includes the studies of various biochemical, biophysical, biophysiological techniques for understanding embryological events at molecular level as well. How the molecule affects the development, how the genes affect the development, how selective expression of genes ultimately lead to the formation of a complete animal. All these can be studied using this chemical embryology. The next branch of embryology is the teratology. Teratology is the branch of embryology concerned with the study of malformations or birth defects that are generally due to the influence of certain chemicals or certain substances, and those substances which cause such birth defects are called teratogens. So some of the examples of this teratogenic development are phocomelia and ectomelia, and many other diseases that are concerned with the development. Phocomelia is the complete absence of the limbs or deformation or malformation of the limbs. The ectomelia is nothing but the development of child without arms. Once we study about the different types of branches of embryology, let us next move on to scope of embryology. What is the reason behind the study of embryology, or why we have to study embryology? So to be very, very specific and precise, embryology is the most important field in biology. It explains the details of ontogenic development of an animal from a single fertilized cell, how a single cell will be transformed into an entire organism. See, even the life of a blue whale, the elephant, the ostrich, everything begins with a single cell. Single cell which is not more than 1 millimeter in length. So even the elephant zygote is not more than 1 millimeter in diameter. So such a small cell will ultimately become an entire organism, such a gigantic organism. How is this possible? It's very fascinating to study about of these creatures, right? So it explains the details of ontogenic development of an animal from a single fertilized cell. It gives basic information about the physiology, genetics, determination of sex, and various diseases, as well as some hints about organic evolution. See how the contact between the cells will be established when they start developing, how the differentiation of the cell takes place, how cells communicate with one another and guide themselves to form different organs, how cells migrate from one place to another place to establish themselves in some other region where they become new organs, how sex is determined, whether it is the chromosome that is defining the sex or the genes that is defining the sex. All these details could be studied using embryology, as well as a very strong evidence for organic evolution could be established using comparative embryology, about which we have studied in the previous slides. Embryology also plays a key role in human welfare. It helps in understanding the causes of congenital malformations, cancer, aging, and in improving the breeds of domestic animals, in controlling pests and vectors of diseases, and the formation of test tube babies as well. So these are the certain applications of embryology. So if we study embryology, then we can apply the knowledge that we have acquired from embryology in these fields. Say for an example, if we study about teratology, then you can easily find the reasons behind the pocomalia, the baby that are born without legs and hands, or sometimes if they are born, they will be having malformed legs and hands. This pocomalia is a condition which was observed during 1960s, where a sleeping pill was used by certain women to induce sleep, and these sleeping pills were found to be causing the defects. That means they were hindering the pathways that are able to produce limbs in babies. And ultimately, the babies were born with malformed limbs and other things. So, after the discovery of pocomalia, the consumption of sleeping pills, a particular sleeping pill, by the pregnant women was avoided. And this was possible just because of the study of the different chemicals that can affect the development of a baby. As well as the study of cancer, and then studies about aging, and very importantly, improving the breeds of domestic animals, dairy animals, poultry animals, etc., as well as this control of pests. If we are able to find out the different stages of developments of pests, then we can easily try to design the mechanisms and methods to control pests as well. The vectors of diseases. See, if we study the development of mosquitoes, then we can easily define and design a method by which we can control the mosquito-borne diseases such as lymphatic filariasis, malaria, etc. And also this helps in ART's assisted reproductive technologies, about which in detail we have studied in the 10th session of the same chapter. So all these kinds of studies are only possible if we have a profound knowledge of embryology and development of the fetus. Some of the latest phenomena such as teratogenesis, cancer, animal breeding, test tube babies and cloning, as well as pest control are the most important fields of embryology. With the success in cloning experiments of Ivan Wilmot in 1996, a new concept of cloning without involving germ cells has originated, which is useful for biological resources as well. The advantages of the cloning of plants and animals are numerous. So this is a pictorial depiction of how the first cloned sheep was produced in the laboratory of Ian Wilmot, and detailed studies of, we have studied about this cloning in detail in the 10th session of the same paper. You can switch back to the 10th session and look into the mechanism by which a new cloned sheep was produced in the laboratory. The high yield food plants such as wheat, corn, rice can be selected and abundantly reproduced. Cloning would give animal breeders a tool for exactly reproducing highly desirable animals. For an example, cloning would make it possible to create 1,000 copies of a prize dairy cow to help feed growing population. Endangered species can be saved by cloning numerous replicas of the best of the few remaining individuals. So these are some of the advantages as well as applications and the scope behind the study of embryology. For any more information about the scope and branches of embryology, you can refer these textbooks. Thank you.